Scientific, and we are excited to have the Thermo Fisher Scientific team join us this morning. We are recording, so we'll send out this presentation along with the slides to everyone after this. If you have any questions during the presentation, please put those in the chat box, and we will answer them at the end. I'd like to introduce our speaker today, Gail Glykoff. Gail has been the Applications Lab Manager for Water Analysis Instruments here at Thermo Fisher Scientific for over 20 years. There's always something happening, new products and technologies, regulatory changes, and evolving customer needs. Visiting customers is one of her favorite activities. Before joining Thermo Fisher Scientific, she had a variety of roles, including oceanography tech and analysis in regulated environmental laboratories, hazardous waste emergency response and investigation scientist, mobile lab and field testing chemist, and laboratory services procurement for Superfund sites. Go ahead and take it away, Gail.
Okay, that's great. Thanks, Paula. All right, well, hello, and thank you to Midland Scientific for inviting me to present this webinar, and thank you for tuning in. I'm glad to be with you here today. Here is our agenda. We will touch on the importance of pH in the various stages of producing fuel ethanol, from starch to sugar, sugar to alcohol, and the final quality of the fuel ethanol product. We will look at how to choose the best pH electrode for your use and the best practices to achieve good performance and longevity of your pH electrodes. Let's get started. The first step in the process of turning corn into fuel involves converting the starch to sugar. This is corn. In the USA, corn is the primary starting material for fuel ethanol production. A corn kernel is composed primarily of starch, and that's what will be turned into fuel ethanol. When starting with dry corn, the kernels are milled and slurried with water to create a mash. Then it is time to add alpha amylase and start the process of converting starch to sugars. This is the alpha amylase enzyme, and it's generally used in fuel ethanol production. It starts the process of breaking down the starch found in grains into fermentable sugars. As you can see, it's a complex molecule. It has a special property that makes it very useful, and that property is pH dependent. Enzymes are catalysts. They're a special kind of protein molecule that speed up a chemical reaction. The enzyme has a complex shape that fits with a specific type of molecule. It works a bit like a lock and key. In our case, starch molecules are represented by the green substrate, and the alpha amylase enzyme is represented by the gray shape. The starch molecule fits nicely into the alpha amylase enzyme. Once they fit together, the reaction is catalyzed and the breakdown of the starch into shorter chain sugars happens more quickly. The shape of the enzyme and how well it works is influenced by pH and temperature. If the pH is not optimum, the lock shape of the alpha amylase enzyme changes. When that happens, the starch molecule no longer fits well with the enzyme, and it is not held in place as needed to speed up the reaction. If the pH is too extreme, the enzyme will be denatured and it won't work at all. If you've ever eaten ceviche, you've seen how the citric acid in lemon or lime juice denatures the protein in the seafood, changing the color and the texture as if it was cooked, just by changing the pH. Temperature is important in a similar fashion. At an optimum temperature, the enzyme catalyzes the reaction quickly and with highest efficiency. If the temperature is too high, the enzyme may be denatured and it won't work at all. Cooking an egg is a good example of how temperature denatures the proteins of the egg. It completely changes the shape and consistency of the protein. That is why pH and temperature are frequently controlled and or monitored at the mash stage. In addition, pH and temperature can control bacterial levels. Alpha amylase enzyme starts the process of breaking down starch by converting it to complex or long-chain sugars, like the maltodextrin sugars shown on the right. This typically happens in the slurry and liquefaction process. In the next stage for fuel ethanol production, the complex sugars are converted to simple sugars, such as galactose and fructose, which are favorite foods for the yeast. One or more other enzymes are added to ensure this process happens as quickly and efficiently as possible. This typically happens before or at the start of fermentation. Let's do a fun fact Q&A. The question is, where else is alpha amylase found? Think about this. Choose any that apply. Certain types of fungus, human saliva, seeds that contain starch, or pancreatic juice. It's kind of an interesting array of options for where you can possibly find this. If you guessed certain types of fungus, you are right. This is a photo of a type of aspergillus fungus on a tomato. While you may not want to eat aspergillus or breathe it, it has a handy ability. It produces the alpha amylase enzyme. If you guessed human saliva, seeds that contain starch, or pancreatic juice, you are also right. The ability of biological systems to break down starch into simple sugars is very useful for extracting the energy from the starch. Now it's time to look at converting the sugar to alcohol. The scanning electron microscope image on the right is a strain of Saccharomyces cerevisiae. Now that's a mouthful. This is the yeast commonly used for converting simple sugars to ethanol at a fuel ethanol plant. Did you know that this is the same yeast commonly known as brewer's yeast and baker's yeast? And it is the most common yeast associated with winemaking, although the winemaking yeast is a different strain that tolerates higher alcohol contents. Yeast is a living organism that consumes glucose and produces ethanol and carbon dioxide. Close to 90 to 95% of the glucose is converted to alcohol, according to the Penn State Extension website. Like enzymes, yeast has a preferred pH and temperature range. pH also can be adjusted to control bacterial growth or to reactivate a stalled fermentation. At the end of the process, fuel ethanol is the ultimate product, and the pHE measurement tells us something about the quality. pHE is the hydrogen ion activity of denatured fuel ethanol and ethanol fuel blends, as measured according to ASTM D6423. The pHE is a good predictor of the corrosion potentials of ethanol fuels. When the pHE of ethanol is below 6.5, some adverse effects can occur. Automotive engine fuel pumps can malfunction when films form between the brushes and commutator. Fuel injectors can fail from corrosive wear, and excessive engine cylinder wear can occur. When the pH is above 9, fuel pump plastic parts can fail. pHE is different than total acidity because total acidity does not measure activity of the hydrogen ions. Total acidity overestimates the contribution of weak acids, such as carbonic acid, and can underestimate the corrosion potential of low concentrations of strong acids, such as sulfuric acid. The two measurements both provide useful but different information, and they complement each other. The ASTM D6423 PHE method is a pH measurement made under specific conditions, including the use of a specific type of pH electrode, testing at 22 degrees C plus or minus 2 degrees, a timed reading, a rehydration procedure, and a defined cleaning procedure. Critical electrode features for pH testing include a high leak rate reference electrode to minimize junction potential drift, especially in extreme low conductivity solutions like fuel ethanols, and a large membrane glass electrode to perform better and have longer life in alcohol fuels. Per ASTM 6423, the Orion Ross SureFlow pH electrode is recommended as having these necessary features. PHE readings are taken at exactly 30 seconds because dehydration of the glass electrode membrane will cause the meter reading to drift. Because alcohol dehydrates the glass, time spent soaking in alcohol solution should be minimized. I participated in the ASTM interlaboratory study in 2014 that determined the precision stated in the PHE test method. During that time, in my lab, we studied some aspects of the PHE method, including this timed reading. We found that at 30 seconds, there's enough time for a fast electrode like the Orion 8172 SureFlow to respond and achieve consistent results. And it is a short enough time to avoid significant dehydration of the electrode, which would prevent good response to hydrogen ions. Immediately after each measurement, it's important to remove the electrode from the ethanol to avoid further dehydration of the glass bulb. Rinse and place the electrode in pH buffer to rehydrate and store in between PHE samples. Following the ASTM PHE method, the electrode is soaked in water-based pH 7 buffer between readings to prepare it for the next sample. This rehydrates the glass electrode, a necessary step to preserve the electrode's response characteristics. The electrodes are cleaned and rehydrated at least every 10 samples by alternately soaking in acid, then alkaline, then acid solution for about 30 seconds each. New electrodes are treated by this procedure before the very first use. During the interlaboratory study, in our lab, we found that this cleaning rehydration procedure is indeed a necessary step to preserve the electrode's performance characteristics. Without this procedure, PHE readings are not consistent or reliable, as seen in this chart. Readings of the same sample can change from less than pH 6 to about pH 8. But with the cleaning rehydration procedure, repeated PHE readings become very consistent and reliable, as seen in the bottom chart. The cleaning rehydration steps greatly improve the performance and the PHE readings, and they're well worth the effort. To see more about testing pH in ethanol fuel, get our application note. Scan the QR code now, or visit us at thermofisher.com and search on Orion SureFlow. Scroll down to the product information section of the web page to get your copy of this application note. Not all pH testing done at a fuel ethanol plant will be done on the final product. In this segment, we'll look at pH electrode features and how we might choose a pH electrode for other pH testing needs. This is an Indy 500 race car. Would we expect to win the Indy 500 with a minivan? Probably not. Can you bring your family on a camping trip in an Indy 500 race car? That's not happening. So different automobile designs have different uses, and choosing the right one makes a difference. Are all pH electrodes the same? They do all measure pH, but different designs offer different features. If we're looking for long use life, easy calibrations, quick response, and accurate readings, choosing the right electrode can make a difference. pH electrodes come in glass or epoxy body styles. Glass body electrodes can be used in almost any sample, are easy to clean, and can handle higher temperatures. Some glass electrodes, like a SureFlow electrode, come with a removable bulb guard to protect the glass pH bulb from damage. On the other hand, epoxy body electrodes have a durable, rugged body that resists breakage. They have a built-in bulb guard to protect the glass pH bulb. So we can choose which body style best suits our needs. pH electrodes come with a choice of junction styles. You may not know it, but the junction style is closely associated with the performance of the electrode, such as the speed, stability, and lifetime. The junction is generally a porous substance that sits between the pH electrode reference solution and the sample. It enables the necessary electrical connection between the reference electrode and the sample. If the junction doesn't work well, the electrode can be slow and drifty. A clogged junction may not work at all. Choosing the right junction makes a difference. Ceramic junctions are a classic choice that works very well for many samples and gives reliable performance. Glass fiber and/or wick junctions are used for refillable or for low-maintenance gel-filled electrodes. They work well for many samples. Junctions like the SureFlow, sleeve, and glass capillary junctions are ideal choices for challenging samples that are dirty or viscous, or have a high or very low salt content. They respond quickly and tend to resist clogging. SureFlow electrodes have the added feature of a quick flush mechanism. This makes them easy to maintain and prevents the junction from becoming blocked. All pH electrodes have a reference electrode fill solution. Some are refillable. Others are sealed and are low maintenance. No topping up, draining, or refilling needed. In general, refillable electrodes tend to make the best electrical contact between the reference and the sample. Because the fill solution can be drained and refilled, it's possible to restore performance to a refillable electrode with timely maintenance. These have the longest life and the best performance overall. Low maintenance electrodes are filled with either a solid polymer or a gel fill. Low maintenance electrodes don't require topping up or draining and refilling the electrode fill solution. They're convenient to use and only require periodic cleaning. Typically, they do not last as long as refillable electrodes. For accurate pH measurements, we need to pay attention to the effects of temperature, because temperature affects the accuracy of both our calibrations and our pH measurements. Temperature effects are especially significant when sample temperatures are hot or cold. For measuring temperature, we can either choose to add a separate automatic temperature compensation or ATC electrode, or we can choose a pH triode, where the temperature sensor is incorporated into the electrode. So how do we decide? Well, an ATC will usually last for years. So when it's time to replace the pH electrode, we can keep the ATC and continue to use it. On the other hand, having a triode means we only need one probe to make the complete measurement of pH and temperature. A triode fits into a smaller container, so we need less volume than if we have two separate electrodes. Triodes come in a limited choice of pH electrode styles. These are the three electrodes that you will see most often in my applications lab: the Orion 8107 BNU-MD, the 8157 BNU-MD, and the 8172 BNWP. They're all double junction Ross reference electrodes, and that means they are fast, accurate, and reliable. They're also quick to respond in warm or cool samples. The 8107 and 8157 are triodes. The 8172 works with an ATC. They're all long-lasting with routine maintenance and proper care. This is my favorite pH electrode. It has a unique glass capillary junction and a Ross gel reference, which makes it a top performer. It's accurate and fast, even in pure water and low conductivity samples. The calibration holds well, and the capillary junction resists clogging. It is a low maintenance electrode. It has a durable epoxy body and a protected glass bulb. It's a triode with an integrated temperature sensor, and it's good for portable pH testing and in the lab. Be aware it's not for use in hot samples. Keep it at less than 60 degrees C or 140 degrees Fahrenheit. Use proper storage bottle technique. We'll talk about that shortly. This is my second favorite pH electrode. Like all Ross electrodes, it's quick and stable, even in cold and warm samples. It's accurate and reliable with an easy maintenance, sure flow, quick flush junction. This junction does not clog. It can always be cleaned and restored. It comes with an optional removable bulb protector. And it's good for lab testing. It's also recommended by ASTM D6423 for PHE testing. Be aware, you do need to use an ATC for temperature compensation. And this is the last of my top three favorite electrodes. Like all Ross electrodes, it's accurate and stable, even in cold and warm samples. It's refillable, so it's easy to restore performance by cleaning, followed by draining and refilling the electrode fill solution. It has a durable epoxy body and a protected glass bulb. It's a triode with an integrated temperature sensor. It's a good choice for portable or lab pH testing. And these are three popular meter options for pH testing. We offer easy-to-use portable and benchtop pH meters, like the Star A221 and the Star A211. We offer a benchtop versus Star meter that offers from 1 to 4 testing channels for pH, ORP, conductivity, and/or ion selective testing. Talk to your Midland Scientific representative about our Thermo Scientific Orion meter kits. All right, so our final segment of this presentation is this: mistakes and best practices for longevity and good performance of our pH measurement system. Have you seen pH electrode problems in your lab? I have. For example, the fill solution is dried out and turned to crystals inside the electrode. There's very little fill solution left in the electrode. The electrode bulb has dried out or is coated and dirty. The fill hole is covered while the electrode is being used to make measurements. Wrestling with an electrode to get it into or out of a storage bottle, or rinsing with water that's not clean and purified. Briefly, here are 7 common mistakes that are made when measuring pH. They include incorrect pH electrode storage, storage bottle mistakes, fill solution neglect, incorrect electrode rinsing, overlooking temperature compensation, using degraded pH calibration buffers, and failure to clean the electrode. These negative effects can occur due to the mistakes. They are shortened electrode use life, unstable pH readings, failure to calibrate, slow pH response, unreliable pH readings, and pH measurements that are just not accurate. pH is a very simple measurement until it's not. Age, neglect, and contamination can ruin the performance of a pH measurement system. A pH electrode is actually a precisely designed scientific instrument. Depending on how we handle and maintain our electrode, it can perform like a well-tuned race car or a junker. So pH electrodes require specific care, maintenance, and handling to properly perform. If the pH measurement drifts, is slow, or doesn't seem reliable, the electrode may be in need of a tune-up to get it back on track. In the next slides, we'll touch on the 7 recommended tune-ups for your pH electrode. For details, scan the QR code and watch this video to learn about the 7 common mistakes that can negatively impact pH electrode performance and what to do instead. This video is posted to our Thermo Fisher Scientific YouTube site and is easily accessed by searching on 7 common mistakes when measuring pH, or by scanning this QR code. Tune-up number 1, correct electrode storage. Use a specialized pH electrode storage solution and close the fill hole before storage. This prevents depletion of the glass pH sensor, crystals clogging the junction, and a shortened use life. Tune-up number 2, storage bottle savvy. Remember to loosen and unscrew the cap from the bottle before removing or inserting the electrode. When removing the electrode, prevent an unwanted vacuum that can pull fill solution out of the electrode and shorten the electrode use life. When returning the electrode to the bottle, prevent unwanted bubbles being pushed into the junction that can stop it from working properly. Because a picture is worth 1,000 words, here's a graphic that shows how this works. Remember to never push or pull an electrode into a closed storage bottle, as this creates a vacuum or creates pressure. When removing the electrode, open the bottle first by unscrewing the storage bottle, taking it off. Slide the cap to the top of the electrode so it's out of the way, or slide it off the bottom to remove it. When replacing the electrode, first slide the cap toward the end of the electrode and then screw the storage bottle onto the cap. Avoid pushing or pulling the electrode once it's sealed in the bottle. Tune-up number 3, fill solution care. If you have a refillable electrode, top up the fill solution before each use. Regularly drain and refill the electrode to keep the electrode functioning as expected. In general, every 2 to 4 weeks is helpful. In your lab, it may be suitable to do this more or less frequently. Low old, crystallized, or contaminated fill solution will cause slow response, drift, and loss of calibration. Tune-up number 4, correct rinsing. Use generous amounts of purified water to rinse electrodes before and after each standard or sample test. This prevents cross-contamination and bias due to memory effects. Tune-up number 5, temperature compensation. Temperature can change the known values of your calibration buffers, and temperature changes the pH electrode's slope. For example, a pH 10 buffer has a known value of 10.01 at 25 degrees C, but it has a pH of 9.89 at 40 degrees C. The ideal slope of a pH electrode is about 59 millivolts at 25 degrees C, but is about 64 millivolts at 50 degrees C. These examples are why we will want to pay attention to the effects of temperature on our pH calibration and readings. We can use either a triode electrode or an ATC with a pH electrode. These will ensure proper pH temperature compensation. When using a temperature sensor with a pH electrode and an Orion meter, during calibration, the meter will automatically assign the correct known value of the buffer at the measured temperature. When the sample temperature differs from the calibration temperature, the Orion meter uses the temperature data from the sensor to compensate the calibration slope with a temperature change. In these ways, temperature compensation ensures that pH readings are accurate no matter what temperature the standards or the samples are at. Here's an example. If I don't have temperature compensation, I have to calibrate and test samples at the same temperature to get accurate results. If my sample's at 45 degrees C, I may put my buffers in a water bath and equilibrate them to 45 degrees C. Then I calibrate pH, making sure that the correct buffer values at 45 degrees C are assigned. When I measure my sample, it reads 5.3. This is a good result. However, if I calibrate my meter at room temperature, for example, 21 degrees C, while my sample's at 45 degrees C, I won't get accurate results without temperature compensation. In this case, my meter would read pH 5.0. This would give me an incorrect pH reading that's too low by 0.3 pH units. If I do have an ATC, temperature compensation will be automatic. The sample temperature will be measured and the calibration slope will be adjusted for that temperature. Now my measurement will give me the correct pH reading of 5.3 pH units. With temperature compensation, I can calibrate at room temperature and get good pH results for samples that are at other temperatures. Tune-up number 6, calibration buffer integrity. Check the expiration dates and dispose of expired buffers. Every day, use a fresh pour from a bottle of properly stored, non-expired buffer. Don't use yesterday's buffers for today's calibration. Prevent bad calibrations because compromised buffers will compromise pH test results. And tune-up number 7, keep it clean. Rinse the electrode and wipe with a lint-free wiper before storing. Periodically clean an electrode to keep it operating at its best. A dirty electrode can drift, become slow, and give unreliable readings, so keep it clean. Well, thank you for tuning in for this presentation on overcoming pH challenges in the fuel ethanol plant. I hope you found it interesting and useful. I'm happy to answer any questions that you might have. Please put your questions in the chat if you haven't already done so. Do we have some questions?
All right, thanks, Gail. That was a great presentation. I do have a couple questions that came through. The first one is, what is the best overall electrode for operator use in the field or plant, and why?
Okay, thanks, Paula. Well, for most field plant applications, I would tend to steer people to our Orion Ross Ultra 8107 BNU MD and a portable meter. As I mentioned previously, this is my favorite choice for quick, accurate, stable pH readings. It is a low maintenance gel pH ATC triode with an integrated temperature sensor. It has the durable epoxy body and a protected glass electrode. So that's convenient and more rugged for field and plant use. It holds a calibration and resists clogging. But if your samples will be higher than 60 degrees C or 140 F, I recommend a different electrode. Low maintenance electrodes with their gel or solid fill solutions, they're not generally the best choice for highest temperature samples, so I don't usually recommend them for that use. For a good refillable pH electrode suitable for field or plant use, I recommend the Thermo Scientific Orion Ross Ultra 8157. That would be my choice for accurate and stable pH readings. It's a triode. It has the integrated temperature sensor. It has the durable epoxy body and protected glass electrodes, so it's convenient and more rugged, but it can also be used up to 100 degrees C. Before use, the fill solution should be uncovered and the fill solution topped up. Cover the fill hole before storage. So those would be my top two recommendations.
Thank you. One other questions I had, if I'm getting inaccurate readings, how should I troubleshoot that?
Right. So when a customer asks about inaccurate readings, they're often concerned about sample readings that aren't reading as expected. And they will need to figure out if there's something going on with their pH equipment or if the sample really does have an unexpected pH value. My first recommendation is to verify the calibration. So get your bottles of pH calibration buffer and check the expiration dates. If they're good, choose the one closest to your sample pH and pour some into a clean, dry beaker and read the pH of that buffer. If the pH electrode responds with a stable and accurate reading of the expected value, then we can see that the pH equipment is good. At that point, I would retest the sample to confirm that the unexpected sample pH is repeatable. If the pH electrode does not respond with a stable and accurate reading of the expected pH value when we check the buffer, then some electrode maintenance is generally the next step. Cleaning is a good choice to start. Then refillable electrodes should be drained and refilled. Soak in storage solution before recalibrating with freshly poured unexpired buffers. Then try testing the sample again. And feel free to call our technical support team at 1-800-225-1480 or email our technical support team at wlp.techsupport@thermofisher.com for more assistance.
Great, thank you. Did anyone else on the call have any questions? If not, like I said earlier, we will be sending out this presentation to everyone who registered so you can review the slides at your own leisure. And I want to thank everyone for their time this morning as well.
Thanks, Paula. Goodbye, everyone. Have a good day.
Awesome. Thank you.